Well, welcome everybody to Hydrotera's latest webinar. Today we're joined by Simon Griffith and Mark Taylor. Simon's a professor from Griffith University's School of Natural Science, and Mark Taylor is our Chief Environmental Scientist at EPA Victoria. So great to see you all here today. The topic is Birds and the Bees, Insights from Biomonitoring for Environmental Pollutants. It's an interesting topic because we're seeing more of this happening where we're looking to monitor what's happening in nature, in the environment, and look at whether there's an analogue between that and our own human health and reaction to contaminants. So it's definitely a growing area, and given the way things are turning up in nature, perhaps a bit of an alarm bell for us to be keeping an eye on. There's a picture of our two presenters. A little bit about our presenters. So, Professor Mark Patrick Taylor has, this is his second webinar he's done with us, so really appreciate your time, Mark. He is Victoria's Chief Environmental Scientist at EPA Victoria. He is also an Honorary Professor of Environmental Science and Human Health at Macquarie University. He has over 25 years of experience in this field with expertise in environmental geochemistry, environmental health, environmental forensics, and citizen science. He has published over 200 peer-reviewed papers and book chapters and has received numerous awards and recognitions for his research and teaching excellence. He is also a passionate advocate for science communication and public engagement. He's joined by a colleague, they used to be colleagues up at Macquarie University. So Simon Griffith. Simon has made significant contributions to the evolutionary ecology of birds, focusing on evolutionary fitness in relation to mating, parental and social behaviour. His research spans various species, including the Gouldian finch and hybridising long-tailed finches, with a key focus on the zebra finch. Simon established the longest wild population study of zebra finches, complementing global captive research. His current work explores the intersection of physiology, environmental variation, and fitness, particularly in response to extreme heat waves and contaminated environments. Since completing his PhD at the University of Leicester, is that how you pronounce it? In 1998, Simon has published over 200 peer-reviewed papers. He began his career in Europe, Uppsala University, Oxford University and Imperial College London, before moving to Australia in 2004 at the University of New South Wales. Since 2007, he has been at Macquarie University holding 2 prestigious research fellowships and a continuing academic position. So really the nub of it is we've got a couple of experts here, one who's more concerned perhaps with human health than Mark, with his role at EPA. And then we've got Simon, who's looking more at animal health than natural systems. And it's really interesting to explore the nexus between those two things in this presentation. Before we get started, we love your questions, so please feel free to raise them in the Q&A, and I will read out your questions at the end of the presentation, and our two presenters will do their best to answer those questions. Why does Hydrotera do these webinars? We believe in sharing knowledge, and we like to facilitate that, and we like to take our leadership position in industry. So without further ado, I will hand over to Mark.
Thank you, Richard, and thank you for having me back again. enjoyed the last one, so hopefully it'll be just as good and enjoyable today. So first of all, just a very quick slide about the organisation that I represent, the Environment Protection Authority, which is Victoria's environmental regulator. Some of you would know that we got a new piece of legislation in 2017, which was uplifted in 2021, and it changed the approach that we were asked to take. And the approach shifted more from the end of pipe, where we assess harms there, to the prevention of harms. And our act is very clear. It says all duty holders have that duty to optimize their activities so it minimizes and prevents harm to the environment and human health. And as part of our, on the right-hand side of this slide, you can see our regulatory queue, and it sort of describes the various activities that we undertake to facilitate our act, covering things from inform and educate and setting standards and monitoring compliance, and of course, enforcing the law. And I'm pleased to say that we have really been using the law to make our act good and to bring polluters to bear their responsibilities, to face their responsibilities. And we have nearly 100 cases in the court at present. But one of the key things of our act is really to inform and educate and to make community central to preventing harm. In fact, the general environmental duty that the EPA has says everybody has a role and must undertake actions to minimize harm. And as part of the work that we do to help the community understand what they can do or where pollution is, we use data. We collect data through a range of programs and through ad hoc investigations, and that is right at the front of the decision making, and science underpins all of the decisions that we make at EPA. Let's go to the next slide, please. So why as an Environment Protection Authority would be interested in biomonitoring? Well, on the right-hand side is a sort of a series of images which really capture some of the things, not all of the things that matter to the community. Birds and bees, which is the topic of today. Everybody that I know has shown an interest in bees. If you produce your own honey, which is what I do, people are kind of fascinated. Oh, you produce your own honey, but people typically enjoy birds, and birds are an integral part of our bees, integral part of our environment. EPA Victoria also has a program that helps people understand the presence and the concentration and the risks and what actions they can take to mitigate harms in their garden from trace metal contamination, as well as improving soil quality. It's called our GardenSafe program. But in addition, as part of the work that we've done as an organisation and the work that I have done along with Simon, we've been looking at birds and bees, and we've used fish and we've used some wild ducks as well in previous work to understand the presence and the dispersal of a range of contaminants in the environment. And we as an organisation are using various sentinel species to help us better understand what risks there are and what actions need to be undertaken. And we're going to talk about some of these, not all of these things, not all of the pollutants that we've worked in, but some of them. Let's have the next slide. So, so really, well, why do we use bioindicators? Well, the slide on the right indicates that intersection between animal health, environmental health, and human health. And beasties sit right in the middle, and they can tell us really about this sort of one health concept, that we cannot divorce ourselves as humans from the animal world or from the environment and the impacts that we have on the environment. And we're able to demonstrate, I think, through this talk today, that really important intersection and the lessons that can be learned by looking at animals to help us better understand what risks there are and what actions we could take. Let's have the next slide.
Just before we do, like philosophically, do you really believe that we couldn't function? Like, I mean, could we not just live with greenhouses and things like that?
Well, okay, well, we could, right? We're talking about doing that on Mars, but that's still an environment. We are the environment. We cannot separate ourselves. The environment is us. So whether it's an artificial environment or a natural environment or a combination of them, that we live in an environment, and even inside a house, for example, it's not hermetically sealed. There are microbes in the house. There are dust mites, for example, etc. They're part and parcel of everyday life. And in fact, some research shows quite clearly that the more closely we are, the closer our relationship with animals, the better our health outcomes are. Whether it's through mental health, through having pets and dogs and cats, etc., or it's through the exposure to contaminants in the environment that helps build up our immunity. And there is emerging research showing that having that interaction is actually healthier than not. Does that help answer your question?
Definitely. Thank you.
That's fine. So where did it all start? Well, it started with two programs that I began with another colleague at Macquarie University called GardenSafe, which looks at and reports on people's trace metal contamination in their gardens. And you can look at all this data on the website mapmyenvironment.com. And we've now been to, I forget the number, it's over 12,000 homes and gardens and inside people's homes. And GardenSafe, the soil program, then merged into DustSafe. And it became a really good platform for engaging the community. And we realized through that work that people's backyards, particularly in the inner city areas of Australia, Melbourne, Sydney, Brisbane in particular, were contaminated with lead. And that comes from two sources. It comes from leaded paint and the use of leaded gasoline. And we then undertook with a former student of mine, Sarah Yazdan-Parast, a program of study to look at the relationship between lead in people's gardens and uptake and harms to those chickens. And we analysed 55 homes. And we analysed it not just for lead, for a range of trace metals. And on the right-hand side, you can access the paper, but you can also access a shorter, quicker version on the conversation. And there's a 5-minute video that I made in the room that I'm talking to you from now that actually sort of captures the story. It's quite a neat little vid. So if we go into we also looked at commercial eggs as well as chickens and domestic produced eggs, and looked at the relationship between those and the sources. Can we go to the next slide? So out of that initial work, which then led into the future work with Simon, and he'll talk about that in a minute, we discovered two things. We looked at the relation to it, soil lead and chicken blood lead. And on the left-hand side, 20, there's no set standard, but 20 birds are a bit more tolerant for lead exposure. And Simon might want to talk a bit about that as he goes through his slides. But 20 micrograms per deciliter is deemed to be the threshold for harms in the birds. And we showed very clearly that in order to keep blood lead of chickens below 20 micrograms per deciliter, we need to keep soil lead at about 166 milligrams per kilogram. That's about half of the Australian health investigation level for people's backyards, which is 300 milligrams per kilogram. On the right-hand side, we showed the relationship there between soil lead and egg lead, and there is no actual egg lead standard. Various people have used the threshold of 100 micrograms per kilogram for egg lead as being a sort of an acceptable number. We could debate that, but that's the value we used. And we were able to demonstrate from the modelling that we needed to keep soil lead less than 117 micrograms per kilogram, nearly 1/3 of the Australian guideline value for residential gardens. So whether you look at the left-hand graph or the right-hand graph, we can see that it's arguable that the level that we use for investigation is probably too high to protect backyard chickens. So we gave this advice out to residents, and this was quite insightful, because it told us that there are real harms in people's yards that people need to consider whether they're going to keep chickens or not. So we go to the next graph, and this is the last one. I'm going to pass over to Simon. We were able to take the GardenSafe data that we collected, and we've done it. We've modelled this information for Sydney, Brisbane, and Melbourne, where we had the biggest density of data. And here I show you on the left-hand side what that plays out in terms of where we have gardens which are safe for chickens versus not safe for chickens. And so what you can see on the left side here, we can see the dark shaded areas are the local government areas where you have soil which exceeds that threshold we set of 117 micrograms per kilogram. And the lighter shade brown is the areas where it would be okay to keep a chicken if you're trying to keep the lead concentration of your egg lead below 100 milligrams per kilogram. And this data matches more or less, not quite, but more or less the information we've gathered for just straight soil concentrations, which is on the right. So the upshot of this is that we're able to take this information and understand what the legacy impacts are, or even in different situations, as Simon's going to talk about, the contemporary impacts from lead emissions and depositions, about choices that we might make in terms of, you know, where to keep animals for different purposes. When it tells us we can also use those animals to give us really important information about potential risks of harm. Now that's enough for me for now. I'm going to hand over to Simon, he picked up the baton with me with a research grant, and we've now gone on to do some really neat studies. Simon, over to you to tell the audience.
Thanks, Mark. Okay, so I'm just going to talk about two aspects of the work today. We're going to talk about linking those humans, linking the human data with the animals that are living in the environment. This is a nice picture of a crested pigeon, a very common urban bird up in Mount Isa. And then I'm also going to talk about some health effects on birds. I'll just give you a very brief introduction to that. Next slide, please. Okay, so I'm just going to talk today about a case study in Broken Hill. That's where we've done most of our work. But I have also worked in all of the contexts that you can see on the slide here. So we worked in Mount Isa with lead and copper. We've been doing work down in Victoria with PFAS. Portland in Victoria with fluoride, and then Theo and Noumea in New Caledonia, where the contaminants are nickel and chromium. So we've done similar work that in all of these places that I'm going to just talk about with respect to Broken Hill. Next slide, please. Okay, so we've focused our work on house sparrows, and the reason that we've done that, house sparrows are actually they're very good birds because they have they've been associated with people for thousands of years, so they live very intimately with people. They're actually called house sparrows because they really prefer to nest in and around our houses and our backyards than further away. So so you can see in that picture on the top, most house sparrows actually live and nest under the eaves of people's houses. Next slide, please. And then the really important thing about house sparrows, which is neat, is that they have a very small home range. So if we go and catch a sparrow in any part of a town, that bird will live its entire life within about 300 meters from the point where we catch it. So essentially what that bird is doing, it's going out and essentially sensing or being contaminated by whatever's in that small home range of about 300 meters. Next slide, please. So that means that our house sparrow is a really good model system for, again, thinking about this One Health idea. It's telling us what's in the environment in that area. We can directly look at the health of that animal, that house sparrow, and the routes to contamination and the consequences of that contamination to a sparrow are probably very similar, actually, to those to people themselves. So we've got this intersection between humans and animals and the environment. Next slide, please. Okay, so case study of Broken Hill. Many of you will be familiar with Broken Hill. Broken Hill they've been mining since the 1880s. And of course, as they did in those days, the town grew up intimately around, in this case, both sides of the mining activity, which continues today. And it's also a very arid area, a lot of dust, a lot of wind, which means there's a lot of contamination throughout the city, which I'll show you on the next slide. So Mark had already done some really nice work, Mark and others actually probably before him. So we have a really good idea of the levels of lead contamination That's what's shown here in this map that's colorized. So we have a good, very nice case study where we can go into a town and we already know what the contamination is like in that urban context there. And you can see some of the levels in the key on the right. Sure enough, the highest or the most contaminated areas of the town are those that are most near the mine and the spoil heaps. So that means we've already got a good understanding of the variability of exposure. And then what we've been able to do, and it's shown in the dots that are superimposed on that map, those dots are actually points where we've been in and sampled birds. And again, we picked birds. House sparrows are very easy to sample, or relatively easy. And so we were able to go and catch samples of sparrows at all those sites. And most of the birds, we would band them, and then we would take a small blood sample and release them. Next slide, please. So most of the work focused on house sparrows because they're numerous, they're easy to catch. A lot of people actually, and again, this link with the community, a lot of people are actually feeding sparrows. So they're very happy to have sparrows in their backyards. And they're also quite happy to talk to us and for us to go and catch the birds that they were feeding. Also rock doves and crested pigeons and white plumed honeyeaters, they represent a range of different species that are very common throughout urban areas in Central Australia. And so this first couple of slides is a paper that we've published, just looking at how well all of these species tell us about the contamination in the areas in which we catch them. Next slide, please. Okay, so what we find in all of these species is that the blood lead levels decrease away from the mining areas. So that means, again, that the birds live in different part of towns. The ones that are choosing to live in the most contaminated areas where we know the soil is most contaminated, sure enough, they have high levels of lead in their blood. And so what that tells us is that all of these species are actually effective, what we might think of as sentinels. So we can catch a bird and they will tell us what is the risk of an animal living in that area. And of course, humans are also animals, so the routes to contamination might be similar. Next slide, please.
Just before we do so,
yeah,
their absorption of lead, is it sort of thermal, or is it from what they eat, or?
Yeah, so actually the next slide, I'm going to talk a little bit about that. And so what we see here, so this is those four species have been split out, and we're looking at the level on the bottom, we're looking at the soil lead level. And then on the y-axis on the left side, we're looking at the blood lead level. Now, what we see here is that the white-plumed honeyeater, which doesn't spend very much time on the ground, the relationship between the soil and the concentration in their blood isn't as clear as it is for these other three species. Now, all of these other three species primarily forage for seeds on the ground. So that suggests that probably they will be, or they will certainly be ingesting dust or lead dust and small particles with their food. So that's one of the routes that they would be taking their lead in. So if you go to the next slide. So again, what that's kind of shown here is that these three ground foraging species are certainly taking in lead differently from those honeyeaters. So they would suggest that these three things are a good sentinel for telling you about the soil contamination in a particular area. Now, of course, one response might be, well, these are birds, you know, people don't necessarily go around foraging from the ground. And therefore, what are these birds able to tell us about the risks to a human of being contaminated? So if you go to the next slide, please, Richard. So that was the next question we asked. We said, okay, sure enough, the birds are telling us what's going on with respect to the contamination in different parts of the town. But does that really tell us anything about the risks to humans living in those same different parts of town? And so the study, which again is published here, that we can make this link available if the slides aren't available afterwards. This is probably the most comprehensive study in the world that's looked at how well animals can act as sentinels for human health risk. So if you go to the next slide, please. Okay, so again, just to be clear about what we've done. So we've used so there's been a program, New South Wales Health have been monitoring child blood lead levels for decades, a couple of decades, and we've used that data. And each one of these small circles on the map these are the sites where we caught the sparrows. We've put a ring through that 300-meter diameter, which is the territory size or a home range size of a sparrow. We've put a ring around each of our sparrow catching sites, and we've pulled up the data for children that have been sampled within each of those sparrow home ranges. Okay, so that means that the sparrows that we were sampling over the last 3 or 4 years, we're comparing the blood lead level in the sparrows, the contemporary sparrows, with the child data that's been collected over the past couple of decades. And I should just very quickly say here that this work has really been driven by an excellent student of mine and Mark's, Max Gillings and Ricardo Ton, who worked on this project with us. Okay, so we've got thousands of child blood lead level data in these next slides that I'll show you, and then we've also got hundreds now of blood lead levels in sparrows. So next slide, please. Okay, so what's really nice here? Well, what's nice is that we can use sparrows. They're really effective at telling us what the risk to child contamination is. So this is the relationship between the sparrow blood in a particular location in town and the relationship with the child lead, blood lead from those same locations. So of course, it's alarming that we have all these children with these high blood lead levels in town over the past few decades. That, of course, is the bad thing. The good thing is that sparrows are a good way of telling us what the risk is. That's important here. Of course, we could go out and measure the soil directly, but they're telling us that being in these same areas as these sparrows, if you've got a sparrow getting high blood lead levels, that means that there's also going to be children in that same 300-meter patch which are at risk of getting high blood themselves, high lead in their blood. So next slide. So we then split the data, and there is some good news here. We split the data to look at the very earliest child blood lead levels, and then the much more recent data. So the sparrow, I'll just be clear, the sparrow data is all collected, it's all contemporary, so it's all collected in the last 3 years. So what we're actually asking here is, do the sparrows better predict what the children were like back in 1991 or today? And you can see here on these graphs, there's a very tight relationship between the sparrows of today with the children of 1991. And what that relationship tells us, it tells us that the children were picking up blood lead or lead from their environment at pretty much the same kind of rate as sparrows. The fact that that relationship was broken down, if we look in the new data from the children of 2020, the breakdown in that relationship is probably the result of both the remediation campaigns and also the intervention programs, where there's been programs, educational programs, reducing the risk of contamination to children. So really simple stuff like don't have your kids sitting in a pile of dirt playing in your backyard. Make sure they wash their hands before they eat their dinner. Make sure they wash their hands after they come off play equipment. Those messages seem to be getting through, and it's broken down that relationship. So that's the good news here. Next slide, please. Okay, so as well as learning about the risk of contamination to people from sparrows, we can also learn about the source of that contamination. So on this slide here, we're looking at the ratios of lead in 2 different isotopes. And at the top right hand, we have the ratio of the lead isotopes in the background, just in the environment in Broken Hill. So that would have been kind of the background environment pre-mining. In the bottom there, we have the ore body. So that's the lead, the isotopic ratio as it's coming out of the ground. And the outdoor dust, so the first layer there, the outdoor dust and the surface soil, you can see is mapping much more closely to the ore body than the background. So that tells us that the contamination in the environment is largely coming from the ore body, in the surface soil and the outdoor dust. Next one. That's also the case for indoor dust. So in Mark's sampling within houses, in loft, in roof spaces, and within the house itself, again, that indoor dust is highly reflective of the ore body ratios. So again, it's telling us that the contamination is coming from the mine. The child blood, so we have a lot of variation in the isotopic ratios. So you can see in that pink cloud of data there, that's the child blood in the 1990s. And if we can measure, add the last layer on, the last layer, next slide, yep, is those are the isotopic ratios in all of the sparrows we sampled. So again, you can see that across town, sparrows have slightly different isotopic ratios, but they're mapping extremely closely with the same ratios in the child blood. So this does suggest, it's very good evidence, that the children are getting their contamination from exactly the same place as the house sparrow, i.e. they're probably getting it in their backyard and in the immediate vicinity of their house. And this isotopic ratio analysis, so we're basically using the blood sample that we take from the bird to measure the isotopic ratios. This can be used in other contexts as well. It's very good at learning about where's the contamination coming from. Is it coming from an ore body? Is it coming from a source from, for example, leaded paint? Is it coming from legacy, you know, the isotopic ratios in fuel? Okay, next slide, please, Richard. Okay, so like the canary in the coal mine, sparrows have been really good at telling us they're capturing the sources and distribution of urban contaminants really well in that context in Broken Hill. We found exactly the same things in other contexts. I'm actually at the moment down in Port Pirie, where we're following up this work. We've found similar things in Mount Isa as well. So we can, in multiple locations, we can go in and look at sparrows, and they're working extremely well as kind of sentinels, canaries in the coal mine. They can definitely be useful to look at that exposure risk in human populations. Anything a sparrow is getting contaminated by, people, particularly children, are going to be getting contaminated by either. Then the last thing that I'm going to talk to you about is actually what's the potential to learn from sparrows with respect to human health. So I was excited, as an evolutionary biologist, I was excited by the opportunity of working in these heavily contaminated towns in Australia, where sparrows have been actually evolving over the past 100 or so years. So in Broken Hill, sparrows have been in Broken Hill for 130 years now. It's been a very, very heavily contaminated environment for that whole period of time. So we can learn about, are animals learning, actually being selected? Are they getting some kind of resistance or resilience to lead poisoning? And we can also learn about what are the subclinical health effects on animals, which again are likely to be also the case in humans. So again, I'm actually sitting in the library in Port Pirie at the moment. Port Pirie is the most contaminated with respect to lead in Australia. And then you can see there Broken Hill and Mount Isa not far behind. And those are the blood lead levels. So a lot of these birds aren't being killed. These birds are actually living and having a lifetime in these contaminated environments. So we can ask questions about what are the subclinical effects? What are the subtle effects that the lead is causing on these birds? It's not just necessarily knocking them out of the sky. Next slide, please. So we've done some genetic work. So in my other work, I do a lot of genomics and genetics of populations. And in this first study that we published a few years ago here, we actually looked at these two environments, Mount Isa and Broken Hill, where we know that birds have had the capacity to adapt to a leaded environment. And we went in and we looked at the genome of those populations, and we said, are there genes in those populations which are at a different frequency to the other populations that we've sampled where there isn't contamination? So the green population showed on the map, they're our non-contaminated populations. and the Mount Isa and Broken Hill are heavily contaminated populations. Next slide, please.
Just before we do, a
Yeah.
genetically independent population.
Yep. So what we mean by that is if there's been a change in Broken Hill, that change has arisen within that population because there's no connection. There are no sparrows and there's no populations of sparrows between Mount Isa and Broken Hill. So house sparrows are so dependent on people that they won't fly thousands of kilometers across the desert. So that means that what's happened in Broken Hill, so any changes that we've seen over the past 100 years in Broken Hill are independent to any changes that we've seen in Mount Isa. And the Mount Isa sparrows actually came from Brisbane. They moved along the road from Brisbane, and the Broken Hill birds actually came up from Adelaide. So they were introduced to Adelaide in the 1850s, and they actually came up on the railway to Broken Hill. So that means those two populations are completely independent. So it means we can study evolution and natural selection in those two populations. They're essentially giving us two kind of different independent views of evolution. Does that make sense?
Yep.
So in the next slide, this is actually kind of echoes that point. So here, all we've got, we haven't got any lead on here. All we've got here is we're looking at some principal components of genetic variation. And we are, look, what this is showing us is that these populations are different from each other genetically. And it also shows us which populations they're closely associated with. So the Queensland populations, like Townsville, Richmond, and Mount Isa, they cluster very closely together. Whereas Broken Hill, as you can see there, it clusters with Adelaide and Port Pirie. Okay. And then next slide, please. Sorry. So the final on that previous study with the genetics, it's not shown on this slide, but what we've found is we've found about 20 different genes that are different in those lead contaminated towns than in other towns. So that we've got genes which are being selected and are helping those birds to survive in those heavily contaminated environments. And when we look at those genes in other animals in which people have studied them, some of the genes have been directly related to lead poisoning in things like lab rats. And some of the genes are to do with stress. Some of them are to do with repair mechanisms for brain. And some of them are to do with visual acuity. All of those things are affected by lead poisoning. So we've got really good evidence. This work's kind of still in progress. We've got really good evidence that's accumulating that's telling us that birds in even 100 years of being in a contaminated environment, these birds are changing. Next slide, please.
Just before we do,
yeah.
So does lead in itself induce a genetic mutation that then get carried on, or is it?
No, it would basically, it's classic natural selection. You got like 10,000 sparrows living in Broken Hill. If one of those birds happened to carry a gene which just gives it a little bit of an edge in that town, then that bird is likely to live longer and leave more offspring than a bird that has the alternate version of that gene. So the lead isn't it's not causing the mutations itself. It's just causing a context in which certain genetic forms will do better than others.
Makes sense.
Okay, and then some of the things that we've been measuring. So again, these birds look very functional. We're catching the birds, they're live, they're going about their daily business. When we release them at the end, they will also return. We are also catching some of these birds a couple of years later. We actually caught one this morning in Port Pirie, in the most contaminated part of town that we caught and measured 2 years ago. So these birds they're thriving in these environments. They're doing okay. But we've been looking at how is lead affecting them in a number of kind of subclinical but quite subtle ways. So we've measured locomotory performance. We've measured how well the birds are flying. And that's going to be important for them because it's going to show how well they can get away from predators, how well they can move around their environment. So lead really strongly affects their performance in flight. From the blood samples we've taken, we've measured the concentration of hemoglobin, and the lead contaminated birds on that graph there, you don't need to look at the detail, but they've got a level of anemia of about 6% higher levels of anemia than the non-contaminated birds, which basically means they're just not got as many blood cells that are functioning well, which means they can't carry oxygen as well. That actually puts a stress on their heart and circulatory system. And in humans, one of the consequences of lead poisoning is actually a higher rate of heart disease, and it would probably be for this reason. So if you're anemic in a lead contaminated environment, that's going to be wearing out your heart at a faster rate. So over a lifetime, that will take years off your life. We've been doing some work looking at differential protein in gene expression. So looking at what are the birds that are lead poisoned at this subclinical level? How are they being affected? So their nervous system is being affected. Again, that's something that is very commonly found in people. Their visual perception, that's also been found in people. So again, what this is telling us, it means we can go into these environments, we can look at organisms that, people might say, oh, well, the birds aren't dying, so the lead isn't a problem. Well, the lead is a problem. It's having these physiological effects on these animals, and it's almost certainly having the same effects on people. People are just like sparrows, really. They've got the same kind of circulatory systems. They've got blood cells that work in the same way. If all these birds in these towns are being affected, there's a really good chance people are as well. The final thing that we've been looking at is with respect to health is in the top there, we've looked at sperm morphology. So sperm morphology is really significantly affected. They have a lot of damaged sperm in contaminated environments. We've shown that with lead, but also with cadmium and also with nickel and chromium. So again, that's going to be affecting reproductive health. So there's a whole bunch, and there's a whole bunch of other areas in which health of animals generally, rats, mice, humans, birds, share very common physiology. So lead will be affecting many of these similar things in people. I think that's last slide, Richard.
Before we move off, a couple of questions. So the total sperm count, did you have a look at that? Like, is it affecting them?
No, we haven't measured sperm count. Sperm count's quite hard to measure in in the field. So we're grabbing these birds. We can take a blood sample, we can take a sperm sample quite quickly, but we're having to freeze that sample and store it for use later on. So we're not looking at sperm count. It might be something we did in the follow-up study.
I always sort of wonder, there's that statistic out there that the sperm count in humans has dropped by 50% in 50 years. Do you think that's a reasonable indicator of a changing environment? Or
absolutely, it would be. I mean, sperm cells are very sensitive to anything that's not, anything that makes an individual not healthy is going to affect sperm counts, because making lots of sperm cells is a physiologically costly thing to do, and it's hard to make them well. So what we do find when we look at the morphology is that we just have a higher percentage of sperm that just don't work. They've either got bent heads or they haven't got tails. So, I mean, clearly that's going to affect their ability to actually function. Yeah.
Can I chip in on that? If you're really interested in that, Richard and everybody else, there's a really good movie it's some documentary, you get it online. It's called The Disappearing Male. And it talks about the decline in sperm. It's an oldie but a goodie. The decline in sperm count since about the 50s. And I remember watching that. I actually watched it on a Qantas flight, and I was like, it was a light bulb moment for me, realizing really what the work I was doing, but the impact and the relevance of that work for the broader health of people. So I encourage people it's a 45-minute movie. It's free. It was made by the CBC, the Canadian Broadcasting Corporation, and it was a bit of an eye-opener. And it talks more around endocrine disruption chemicals, but it's the toxicity of all chemicals to which we're exposed have subclinical negative effects. And we still do not know why sperm count is falling, but it is falling, and there is an estimated zero time. And let's hope not, because I like to think some of us are useful. But it's a real thing, it's a real thing, and it is not unsurprising that the insults that we're receiving from the plethora of environmental chemicals are having effect. It's just that those effects are subtle and subclinical.
So the next question I guess I have for you is, given the evidence of all this decline in total sperm count in humans, is there a way of sort of back calculating against the plethora of contaminants that we have out there to work out which ones are causing most impact, or is it just too hard to do? To prioritize what we should be focused on, I guess.
In a very broad brush attempt, you could do that. But what I've said in other conversations is that despite the exposure to an increasing number of contaminants, and there are multiple studies that demonstrate the almost exponential increase in contaminants, including microplastics, humans are living longer and healthier on every continent. But that doesn't mean to say that more fragile members of society aren't affected. But so I think, you know, the absence of evidence isn't evidence of absence of the insult from chemicals, but population wide, we are living longer and healthier than before. But unpicking your question would require a bit more work.
Yeah, no, I'm figuring it's a fairly broad scope. The definition of healthy, surely it's got to take into account ability to reproduce. And if you say that video's got this end point, well, then we're kidding ourselves. We're just living longer, but there's, you know, we're going to stop breeding, aren't we?
Eventually, that wouldn't be a bad thing.
Oh, might be one. Anyway, worth pondering. Just how do you measure the performance of bird flight? I was kind of intrigued by how
so
one does that.
It's a very simple one. So once we've finished doing the other sampling, we literally just release them from the hand, and we've got a 3D. We've got a set of 3 video cameras. They're actually GoPros, and then you're measuring at high resolution, high speed. We're measuring how quickly they get away. So in that graph at the bottom, that's actually the trails of each bird. And in that, we're really just measuring how fast they can go in, I think it's 0.5 of a second. And what that's actually capturing, it's capturing how well their muscles are working to generate force. So we're actually kind of looking at the force that an individual bird can generate. We've also done some work looking at mitochondria, how that's affected by lead. And the mitochondria are the organelles in all of our cells that fuel all of the things that we do. They produce the energy that our bodies use. Mitochondria are also severely affected by lead. So it just means our ability to make fuel from the food that we're eating is also constrained by inner leaded, or when we're contaminated with lead to a subclinical level. So again, that's just going to wear us out. It's wearing out our internal machinery.
Is the productivity of Port Pirie lower than the productivity of Melbourne because of the lead levels?
Yeah, so we've only just, so this is our second sampling trip down here. So we've got we've literally got our lead data. We know that the lead levels are very high in the birds down here. We haven't done all of these follow-up studies yet, but we're literally collecting samples like as I speak this morning and tomorrow. Okay. I think that was one of my slides. I think Mark's next if you advance the slide.
Before I start rattling on, are we finishing at 1:30 just to check?
Yeah, we do Q&A. Yeah. So, Q&A typically goes longer.
So we'll go beyond 1:30. I'm just checking so I can trim the slides.
Yeah, no, that's all right. I've got a bit of a tradition of going over time. Yeah.
That's a self-admission of verbosity. So moving from the birds onto the bees. So it's part of the work that I've done, not just me, but also with a bunch of other colleagues from Macquarie University and elsewhere, but particularly with my students. We've looked at honeybees as biomarkers for environmental pollution. And one of the studies that we've done some work in Broken Hill, of course, we've done some work in Broken Hill and Sydney, but also in New Caledonia. And as a regulator, we've also been looking at bees in and around the Melbourne area for a sort of a related but slightly different purpose. And so I was fortunate to be funded in 2019 to go on a sabbatical to New Caledonia, and where which was a bit of a fun trip, I'll say, as a sidebar, that, you know, I speak a little bit of French and I'd never been there before, but I had to run a program and engage with beekeepers over there who were unbelievably enthusiastic, and they really helped me facilitate access to their beehives. And so what we did there, New Caledonia, you'll have seen in the previous slides that Simon had mentioned, and he didn't really talk about it, but in Namur, which is the capital city of New Caledonia, there is a major nickel smelter, where nickel and chromium are emitted from that facility. And very little work has been undertaken in the city of Namur to understand the deposition and the harms that may occur from those emissions. And we did a parallel study looking at soil and dust, and then we did this other study when we looked at bees. And so I'll just talk a little bit about the bees. Oh yeah, next slide, please. I'm trying to do the slides. So what we ended up doing is bees are really fantastic. They have a different footprint to the 300 meters range that Simon talked about. Bees are a bit like people, they're a bit lazy. If there's a supermarket around the corner, you know, you could have an IGA at the bottom of your units, which is about, you know, 20% more expensive than the Aldi, but the Aldi is like half a kilometre away. So most people just go to the IGA, right? Because there's food there, they can get it. It's the same thing with bees. If they're plants locally, they'll go to them, but they will go up to 9 kilometres is the estimate. But typically, bees will go on average about 2.5 kilometres or so from their hive. And that's what we use here with this figure you can see on the right-hand side, the hives that we've got, which are in the sort of yellow dots. You can see the dashed line around all of those hives, which shows that we're able to cover the whole of the island as an estimate of contamination using bees. And so we ended up with sampling over 1,100 bees, 15 hives, took 20 honey samples, and we analysed multiple soil and dust samples in order to get an understanding. And really, ultimately, the focus was looking at the impact of nickel smelting. So let's have a look on the next slide. So what we have here is a range of plots, and I'd like to thank Max Gillings, who was mentioned earlier, for helping me with these terrific graphs. And you can see on the left-hand side, you can see the concentration ratio between the bees and the honey. On the left-hand side, we can see for boron, it was actually higher in the honey than the bees. But for everything else, all these other elements we've analysed, including lead and including nickel, we can see the concentrations were much higher in the bees than the honey. And that's good news if you're a honey producer, because it means that the honey that you get, even in a contaminated place, which you've showed elsewhere in Sydney and Broken Hill, is actually largely free of contamination. And without going into the details on the graphs on the right, which will be hard for people to see, in essence, the concentration of the element of nickel in this particular case is like an order of magnitude lower in the honey than what it is in the bees. So what you can see, if you just take the big view picture of these graphs on the right, we have on the left-hand side, we have on the vertical axis, we've got concentration, milligrams per kilogram, and on the y-axis, we have we have distance from the facility. And what you can see, whether it's cobalt, whether it's chromium, or whether it's nickel in bees, or whether it's nickel in honey, there is a clear, distinct decline with distance from the smelter, 0 being the nickel smelting facility at a place called Doniambo, and distance from that. It's the same pattern that Simon has demonstrated with the sparrows, with distance from the pollution source. No matter what species you looked at, you can see a really clear spatial relationship between proximity, higher concentrations, and distal, lower concentrations. Oh, next slide, please. So the question that we obviously knew pollutants were getting into the bee, but the question that we didn't really know, that where is it in the bee? And there have been some studies from Italy that have argued that the surface of bees would be really good for looking at ultrafine particles. The capturing of the particles on the bee can be used to use them as sort of markers. The middle is some scanning electron microscope images of the surface of the bees. We didn't do an enormously thorough study. We did a bit of a look-see, but I couldn't see anything on the surface of the bees. Now, on the left-hand side, what we did with the bees that we captured, to help us try and understand where it is in the bee, we took some bees and I split the samples at the different sites that I had, and I did a washing protocol on one half and an unwashed protocol on the other. And on none of those samples on any of the elements, we were able to demonstrate there was a significant difference between washed and unwashed bees. So that made me think, well, it's clearly not in the bee. We can't see it on the bee and we can't wash it off the bee and then see a difference afterwards. And then what I wanted to know was, well, if it's in the bees, what's happening? Is it accumulating over time, etc.? And so on the right-hand side, as further evidence to understand it's in the bee, not on the bee, we looked at the difference between live bees and dead bees. And we can see nearly in almost every case, the blue bars you can see there are significantly different for the same matched element to the orange bars, which are the live bees. So the dead bees are in blue and the live bees are in orange. And in each and every case, the dead bees contained higher concentrations at a significant level, than the live bees. And on top of that, we were fortunate enough at one particular site, and the graph's so small I can barely even see it, but these little dots you can see towards the bottom half, they're like little crosses, the bottom half of that graph on the right-hand side, that was a single sample of a queen bee that we were able to analyze. And you can see that again is even lower. This tells me, when we wrap all this information up, this tells me the following. The queen bee cannot feed herself. She is fed by the other bees. So if she was getting contaminated, the other bees would be passing those contaminants into her, and she lives longer than most bees, which live up to 2 to 4 weeks. Therefore, when we then look at the live bees and the dead bees, what must be happening is the bees are accumulating the contaminants over their lifespan. They're taking it into their honey gut and it's not coming out. And we can see that by the difference between the live and dead bees. But the fact that it's not coming out is evidenced by, albeit one sample, on the queen bee. But the fact is that the dead bees, that's also supported by the fact that the dead bees have a higher concentrations than the live bees. So ultimately, it's in the bee. We were able to demonstrate, and I think going forward, sampling dead bees is possibly a lot better as a pollution indicator than sampling live bees, which also is a bit tricky. And I have now, having owned bees, I've now become allergic to bees, having been mauled by a cranky hive, which is great for a beekeeper. But it actually helps us understand what are the best types of samples to collect. So it's in the bee, it's not on the bee, and it doesn't get into the honey. So it's a kind of a good news story for producers. Can we go to the next slide, please? And then we also then used the same sort of process that Simon's talked about. We looked at the lead isotopic ratios of samples in the bee and some of the samples that we know associated with the smelter slag, the background soils, and deposited dust. And we analysed a mixture of live and dead bees, and they're captured in the middle of that graph. So if you look in the middle of the lead isotopic graph, you can see like a grey envelope that's deposited dust, and that characterizes the isotopic fingerprint of the deposited dust. But the blue squares and the orange circles, they are the live and dead bees. And you can see that they're different to the historic use of leaded petrol, French leaded petrol in Namur, and they're a little bit offset between the smelter slags and the background soils, and that's what you would expect. But the fact that their isotopic composition mirrors the dust deposits, which we know can be linked to emissions and depositions, which also have a particular lead signature. We know that, therefore, that it's the smelter operations which are influencing the outcomes in the bees. So if we go to I think the next slide, and look, part of the broader work I was able to take to the EPA was the use of bees for surveillance. One of my former students, actually a couple of former students, have gone to work for EPA Victoria, and we're able to bring this with us, this sort of intelligence and understanding of how we can use bees along with some of the other species that we look at to trace things like perfluorinated chemicals in Melbourne, and we can see them in some of the bees, albeit at ultra low levels, nanograms per gram dry weight. You can see on the right there, map of Melbourne, Port Phillip Bay, and a different distribution. We do know from other work that urban environments are more contaminated with perfluorinated chemicals than regional areas. That's very clearly established. And then, but also on the left-hand side, another study which was carried out by Cara Fry, a former master's student of mine, she's also at EPA now. She's a health risk assessment worker at EPA. She looked at bees in Sydney and the use of those bees for antimicrobial resistance. And she showed very clearly that class 1 integrons, which originally came from human bacteria, are now disseminated by human activity through antibiotic use, which living in close proximity to bees are then being passed on to the bees. And indeed, what we can show that the presence of the class 1 integrons and their associated resistance gene cassettes, such as metals, antibiotics, indicates very clearly that humans have been important in terms of mediating the DNA transfer into the environment. And the use of keystone species like bees allows to understand the prevalence. We showed in the study, which was also published in Environmental Science and Technology, that antimicrobial resistance was found in every single hive that we looked at in the Sydney Basin. That also therefore tells us that we need to be really aware and cognizant of the use of use of antibiotics and the disposal of antibiotics, because clearly it's permeating every facet of our environment. Which brings me right back to this point that Simon was making, that we are not detached from the environment. This allows us to use the species to understand that one health concept. The environment is us and we are the environment. We need to be cognizant of the pollution that we create and how we deal with waste products before we just unwittingly and unconsciously distribute those into the environment. Last slide, I think. So what have we learned from the work that we've done, both the work that Simon has done and I've done? So we did a piece of work which we published, and Cynthia Isley, now Barlow, published back in 2022, where we actually evaluated some of the citizen science work that we've done and what people got from it. And it was very clear that people are deeply engaged in science. People want to know, even if there is pollution, and they want to know how they can address it and reduce their risks of harms. But working with the community gives us unprecedented access to environments that we normally couldn't reach as a scientist. But we're working with them and answering things that matter to them, birds, bees, for example. And as a scientist and as a researcher, we're able to gather really detailed critical information at spatial concentrations that we couldn't normally access. And it gives us really detailed information about spatial distribution of pollutants, what those risks are, what we should be doing in those different places. Consequently, EPA Victoria continues to use birds and bees for assessment for a multiple range of pollutants, PFAS, trace metals, and pesticides, and also for general surveillance of emerging contaminants. It's a relatively new area to sort of broaden the church from the typical fish analyses that we have been doing at EPA for many years, and a little bit around the ducks. So we're reaching out, we're using it to give us information and intelligence and insights about what to do and what to be concerned about. That's it from me, Richard. Thank you very much indeed.
Well, thanks very much, Mark and Simon. That was excellent. I'll move to the questions. Mark, you able to hang around, and Simon, to answer these questions now?
Yeah, I can hang around, but I've literally got to head off to Broken Hill now. So if I could just answer my questions first very quickly. There's a couple in there.
All right. There's probably a few in the Q&A too, but maybe just have a look at these and then
Yeah.
get on your way. Information about PFAS in bees and hive products. Is that one for you?
That would be Mark.
Yeah.
Microplastics.
So the first one, we haven't finished the study and we're not ready to publish it, but we will. And EPA Victoria has, you know, a clear mandate to make our information publicly available. It's just not finished. But there has been other work conducted, I understand, in New South Wales, in and around the Williamtown area, but the detection limits were actually quite high, and they demonstrated there was no PFAS in the bees, I do believe, but I don't believe the concentrations were low enough. We worked with University of Queensland, and we got much lower detection limits, so we're able to get a much more granular picture. But I think it'll be the same as the trace metals. I wager that it's in the bee, but not in the honey, which is a good news story, if you like. It's not great for the bees, obviously. It's the same thing that other people, Andy Barron at Macquarie University has done a ton of really, really fascinating work about the impacts of trace metals on the neurocognitive capabilities of bees. And so it's not unsimilar or dissimilar to the sort of inferences that Simon has made about the pollution on sparrows, for example. Microplastics absorbed by honeybees, I do not know. Do not know the answer to that. There has been some work about microplastics being on bees, but I think it'd have to be nanoplastics, not microplastics, because, you know, I think microplastics are, by definition, they're less than 5 mil, and I think greater than 1 micron, if I remember correctly. And that's possibly a bit too big for a bee, possibly. Do you want to have a go, Simon?
Yeah, sure. So I can just answer a few. Can I answer a few from the Q&A thing, Richard?
Sure. All right. We can.
So the first question at the top about the itinerant nature of the white-plumed honeyeaters, that's absolutely right. And so one of the reasons that we focused on sparrows is that they're very sedentary, and they live. We know from, you know, decades of work in Australia and around the world that they stay and they spend their whole lifetime in that small area. Absolutely, native birds will typically move through environments to a much greater extent. So that's one of the reasons why that relationship isn't as strong. But again, the other consequence here is it just actually means that the limits, even within that Broken Hill or Port Pirie, you might have quite a relatively small area of contaminated environment, but some of those birds are going to be picking up contaminants and kind of then moving far and wide away from that. So it is an interesting question. You're absolutely right. Yes, they will move around, and which means that we're sampling.
So Simon, let's just I don't think the guys at the other end can see the question, so I'll just read it out. So that first one was, how itinerant are white-plumed honeyeaters? Most honeyeaters follow tree blossoms and may not be in any one area for long. So what was the next one you wanted to answer?
There's another one about yeah, so the broken so it's the one from Simon Sharp at 120.
Okay, just let me get to that. It's like a bit new for us here right now. 114, 116. It's a little bit annoying the way it moves. Can you read it out?
I can read it out. If the Broken Hill study compared soil concentrations to lead in child blood concentrations, how did the strength of prediction of child blood from soil compare with that from sparrow blood? So that is absolutely a great point. So we haven't directly done, or we have looked at that, but only kind of indirectly. So of course, we don't have the soil measurements from exactly the same place as the child historic measurements. And of course, a lot of the remediation work. So if you get a child in one particular house, you might, that particular environment that that child lives in. So one of the reasons that that relationship has broken down more recently is it's a question of where you detecting the soil. So where are you measuring the soil lead? If you measure it just in that home, you might get a stronger reading. But again, what the sparrows are doing is they're measuring over that kind of 300-meter area. So they're kind of averaging the contamination over a much broader area. And of course, that is related to what the child is exposed to, because it's not just the soil in their own home, which might be quite low. You get a dry and windy day, and you'll get a lot of airborne lead from the yards and the streets around that child's home as well. So it's a really interesting question, and it's something where I think the sparrows are good because they're kind of averaging in the way that if you just go and measure a few soil samples in a street, you'd have to measure an awful lot of soil samples to give you that kind of average. And it's also what's mobile as well. What the sparrows are detecting is what's actually mobile and that an animal will be picking up. So hopefully that answers that question.
Can I chip in on that? It's a good question, but the soil is really a proxy, and it's a much more direct exposure route for the birds, and it's a sort of a rough proxy for people. And the reason why I say that is because we did a piece of research looking at dust versus soil lead as the primary source or route of exposure, and it turned out to be dust. And I'll give you a very quick explanation of why that has to be the case in any case. So when kids are born, their blood lead is about the same as the mum's. And at the moment, mum's blood lead in Broken Hill, and according to the last Broken Hill study, is about 0.6 micrograms per deciliter. It's just at now normal population levels. But after birth, children's blood lead rises almost inextricably. Now, kids between 0 and 12 months of age, particularly between 0 and 6 months of age, are sat either in the cot or they're on mum's breast or some other type change table. They are not rolling around in the garden dirt. Therefore, the garden dirt itself cannot be a direct source of exposure. It has to be indirect for kids. But what Simon's work has showed, well, my work and Simon's showed very clearly that the soil is a very good proxy indicator, along with the birds, obviously, of relative exposure. But it provides, in terms of for little kids, it has to provide an indirect source through the remobilization, which would also be added to by atmospheric dust levels. Now, obviously, the behavior of birds is different from people. Another example of that would be during the leaded gasoline era, right? There was a ton of stuff coming out of the tailpipes. That stuff is still in people's soils. But during the leaded gasoline era, kids presented with elevated blood leads. Today, they don't, because they're not rolling around in the dirt in the same way. But the sparrows are still contaminated. There's just a different timing and a different exposure routes.
All right, we better go on to Simon's. Simon, is he still with us?
Yeah, I am. Yeah, I don't think there's any more questions for me though. I think Mark can.
Okay.
Thanks.
Well, thanks very much, Simon, for your time.
Thank you.
We'll let you
thanks.
get driving. But
Thanks very much for
Mark,
your attention.
Okay.
Great stuff.
All right. Thanks very much. I'm going to leave you.
Thanks, Simon. I'll talk to you.
All right.
So,
thanks.
Next question from Matthew Landos. Presently, biosecurity measures and legislation focus solely on pathogens and pests. Can you see a pathway to reform legislation such that the impacts on health resilience of a myriad of contaminants, metals, pesticides, industrial chemicals, PFAS, and plasticizers? Consider avian influenza as an example. Could future biosecurity invest in environmental decontamination as a health measure to reduce disease entry, expression, and impact?
I'm probably not the right person to answer that. That's a real pass back, isn't it? I don't sort of see that we'll be doing environmental decontamination in our urban environments in a great hurry. And the reason is we'll focus on the prevention of the emission of new contaminants, or and in terms of in people's homes, typically because it's private residences, it becomes a difficult space for regulators to work in. But look, you raise a really good point. I think, you know, our cities are industrially contaminated locations just by default, just because of all the activities, vehicle driving, leaded gasoline, former industries, etc. But we need to sort of, you know, we need to look at how do people get exposed, and typically that is through ingestion, which means it's really critical that we need to make sure water supplies are clean and are free of contaminants, or the contaminants that may be present are at levels that do not present a risk of harm. And Matthew would know that we use a risk assessment process to determine what the acceptable thresholds are. That's how we work here. I know it's a bit different in the States. And then the other one is, you know, in terms of food, the Food Standards Authority did their survey of PFAS in foods, I think a couple of years ago, and it showed very clearly, I'll just pick on PFAS, and it's the same with trace metals. We were lower for PFAS than China, the United States, and Europe. So yes, there was a few places, a few types of food that did contain some small, very low concentrations, deemed to be by others to be non-significant, but you know, we're still better off than other parts of the world. So I think actions have been taken to ensure, to reasonable efforts, that food and water is free of contamination. Of course, there are issues when people would argue that's not the case, or samples are revealed that that's not the case, and we know there are some live examples to that in and around the country. But overall, I think we live in a pretty clean environment relative to others.
So taking all that on board, we could debate that till the cows come home. But I think Matthew's got a good point here, right? So we keep pouring more and more contaminants in, like we might call them products, right?
Yeah.
And we keep saying, sure, let's do that. Let's bring this in. We don't seem to really have a process in the world for being able to predict the impact on these things until the genies come out of the bottle. And so then you're doing these studies on bees and things, you know, like we've been doing lead since the 1880s, and now we've been doing plastics since the 40s. Perhaps we could learn from this and say, wow, look, there are impacts there now, but we've got a bit smarter, and yes, there are these impacts, and yes, we can measure them. So I think what the point he's saying is, well, can we bring it forward in the process, right? Can we actually stop bringing more in to the funnel, if you like, and let's get a bit more sensible about controlling that end of it, because you'll never catch up, right?
And that's incredibly hard, because as you would know, Rick, so the ICHEMS process is designed particularly to do that, focusing on the Stockholm Convention listed organic chemicals. And you know as well as I do that, and we got the Minamata Convention. We know on both those cases they're dealing with, you know, the situation of the cat that's got out of the bag somewhat, and we've identified those, and point taken. But at least, you know, the government's eye is now on addressing products, in effect, chemicals that are embodied in products into the future coming into the country. I mean, the challenge that we face is that, it's not my area of expertise, but industry is producing different versions of sort of similar chemicals at a rate that nobody can keep up with, and that makes it really challenging. And I'm sure the argument would be made that, you know, we do need these pesticides, we do need these herbicides, etc., because of the volume of food that we have to produce, because of the world's population is increasing. And so we end up with kind of a bit of a circular argument. And it's a bit out of my space to make that comment, but to sort of give a very sort of research comment on it. But I think the key thing is that we need to be cognizant as a society as a whole about and asking for what's in these products, and for also us as consumers in demanding different and better standards of the products that we buy, and ensuring that, you know, we know where they've come from. Peter Singer's work around understanding the source of products, that focuses more around the treatment of animals in the production of food. Is a really good example that, you know, he basically asked, you need to know, you know, what's the genesis of that food and how it's being produced, and it would be the same thing in understanding about, you know, what's in that food, what additives have been used.
All right. Matthew, that was a great question, I think.
I'll talk to him later.
No doubt, food for thought. Matthew's got another. Oh, Matthew's had a busy day. If interested in sperm counts and effects, this is a useful assembly of data and contextualized in financial implications to society. There's a link there. We'll put it in our next newsletter, I think, but we'll circulate that one around. One more comment from Matthew. USA human longevity has reduced over the last 3 years. There you go, Mark. The data has changed direction and rates of disease are increasing, including diabetes, heart disease, Parkinson's, ADD, infertility, and numerous cancers. I'd question that we are living healthier. Births are down in almost every country except sub-Saharan Africa. Might have to move there. Mark, do you want to respond to that comment, or?
I'm not across the nuance of all the statistics, but birth rate is influenced by other factors as well as fertility. And I know, at least in this country, heart rate disease has been declining. I know there are some shifts in Parkinson's and diabetes in different parts of the world. I know ADHD diagnosis is on the up. We don't really know why, but whether it's just because people are doing more diagnosis, again, that's the other question. But look, I don't deny those comments that there is some emerging evidence, particularly around childhood cancer rates and mortality rates, which have been increasing. There's a paper out recently, New England Journal of Medicine. I think Philip Landrigan was an author, and Matthew might be aware of it. It's worth a read. But, you know, these are all fair comments, and I think, you know, birds as well as people we have opportunities to sort of follow those trends and understand what's happening. Understanding what the causation is another question, more difficult question. But I understand what Matthew's interest is here, and he makes fair points.
Thanks for that. Okay. Mark Dobrowolski. In New Caledonia, much of the native vegetation hyperaccumulates nickel. How did you separate the natural nickel contamination from the smelting works sources?
Well, the answer is we didn't, and we didn't really sample veg. But I think what he's alluding to there, Colin, is that because the plants are hyperaccumulating it, that would then get up into the nectar and the pollen. What I would say is, on the island of New Caledonia itself, actually, they are not in itself. There's no nickel sources per se. It's quite low nickel in the surface and subsurface soils. So maybe that wasn't a relevant factor, but I think to counter that, we could really see a clear spatial pattern in the soil and dust studies that we did, and we saw the same sort of pattern being played out in the bees as well. So yes, it may have had an impact, but the concentrations of nickel in the natural subsoil were relatively low versus other parts of New Caledonia. Good question, though. Thank you.
Colin Farrelly, is the difference between dead bees and live bees simply an indirect measure of the age of the bee? Could the age of the sampling device, bees or birds, be used to improve the signal?
So, yes, the concentration is a function of the age, because the bees accumulate it as they age. So as they're going out foraging and coming back again, they're accumulating contaminants in their body over time. So it is a function of age, but the purpose of me doing the live dead bee analysis, Richard, was to understand, you know, where is it accumulating? As part of the question of understanding where is it accumulating. And I traditionally or previously sampled live bees, but I also went to look at dead bees. But it told me very clearly it's accumulating as they go out. So that's telling me it's, you know, it's not on the body, but it's partly due to their interaction with the environment.
Okay, Simon Sharp. Thanks, Simon and Mark. An excellent presentation, and appreciate your answers and fascinating insights highlighting the benefits of biomonitoring with these case studies. There you go, Mark.
That's very
Keep up
nice.
the good work. Matthew Landos has put a link in there, and he also has expressed his appreciation. So that brings us to an end of the Q&A, Mark. Thanks very much for that. That was excellent. Certainly raising a lot of questions, isn't it? Really appreciate you putting this on. We didn't quite get through the early bird questions, so I might just finish off on those. Can microplastics be absorbed by honeybees?
I mean, nanoplastics likely, but what the
That's right.
don't know. The third one, hundreds of hives used for pollination, has the impact on birds being studied, temporary mass increase in food and chemicals. I don't really understand that question. No, I don't really understand that. The second one, the fourth question, sorry. Did I hear Susie Reitman say that based on the research that HILA for lead is generally protective of chicken eggs? You can draw your own conclusions. We've published our data. You can, as I say, I just provide the conclusions of our study as a factual basis. And then the last dot point, the fifth one, is EPA using technology such as AgriSound to quantify pollination biodiversity. Now, the answer is no. It's not really our space. That might be something more to do with the AgVic, but thank you for highlighting that as a technique. I think that's it for me, Richard. I'm exhausted. I need some lunch and a cup of tea.
Sounding a little weary. But Mark, thanks very much. Really appreciate your time today, and I'm sure everyone else appreciates it too. So thanks very much.
Thank you for inviting me, and I'm happy to do it again some other time.
Excellent. Thanks, Mark.
Cheers. See you. Bye.